College. Originally from a small town in Utah, Bob earned his BA in chemistry at Pomona College and later his PhD in biophysical chemistry from Stanford University, where he studied fundamental biophysics questions related to membrane fusion, the molecular process which underlies synaptic transmission, sperm egg fertilization, and enveloped viral infection. Bob then worked as a postdoctoral research scientist at the University of Virginia School of Medicine and as a visiting scholar at Stanford University, where he studied the membrane binding and fusion of influenza and Zika viruses. At William College, Bob's research lab studies the biophysical chemistry of viral infection using both wet lab and computational techniques. Outside of his work, Bob is the happy and tired co-parent of 3 young children and not infrequently transforms himself into a terrifying ice dragon named Ice Fang to engage them in battle. I will now turn this presentation over to Dr. Rawle.
All right, thank you. Let me just double check. Can one of the panelists tell me, can you hear me okay?
We can hear you. Thank
you.
Okay,
perfect. Yes, thank you for that introduction. I do sometimes transform into an ice dragon. I promise not to do it during the course of this talk. I'm very excited to be speaking to you today on this topic. I'm currently in my office, as you can see, at Williams College, sort of a muggy, rainy day outside. I hope that wherever you find yourself, that you're going to have an enjoyable time during the conference today. So as was said, I'm going to be giving you the science bootcamp session on virology. And this is, from the top, not an easy task. Virology is an enormous field. It touches on many, many, many other fields, and there's really no way I can cover everything. So my goal today is really to give you a framework to think about viruses at the molecular and cellular level, which is kind of where I spend most of my time thinking about. Even still, pretty much every slide I'm going to put up represents an entire subfield, so that's just the nature of the beast. Now, in preparing this talk, I have assumed a basic science background, especially with biology, but essentially no background in virology. So that's kind of the level that I'm shooting for. By the end of the talk, I hope that you'll be able to grapple with concepts such as viral infection, antiviral drug treatments, disinfection, and so on. Throughout much of my talk, I'm going to be using the new coronavirus as a case study, and that's why I've put up this really stunning structural model. It's a zoomed-in structural model of the new coronavirus, which comes from a group called Visual Science. Now, the terminology can be a little bit confusing, so the official name of the coronavirus is SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus Number 2, kind of a mouthful. It is the virus that causes the disease COVID-19 in the same way that HIV is the virus that causes the disease AIDS. Before we get too far in, I wanted to give a quick overview of my research. This was briefly mentioned at the top, but I'll kind of talk a little bit more about it now, just so you have kind of a sense of where I'm coming from. So as was said, I'm a relatively new professor here at Williams College. I am a biophysical chemist, which means that I use tools in chemistry and physics to study questions in biology. There are 3 main projects shown here that I work on in my lab. The first 2 areas relate to understanding viral binding and fusion, which are at the heart of the early stages of viral infection, and I'm going to talk a lot more about this going forward. In my lab, we study these in wet lab experiments using microfluidic devices, fluorescence microscopy. We also study them using mathematical modeling. And I'm going to be talking at the end of my talk about a brief project that relates to both of those things. The viruses that we study in my lab are West Nile virus and Zika virus and Sendai virus, which you probably never heard of, but which we think is super fascinating. The other area in my lab is not directly related to viruses, but looks at membrane compositions of model membranes that are used in drug delivery and drug formulation. So anyhow, that's kind of the 60-second overview of my research, so you kind of know where I'm coming from. One thing that I'll point out is that I am not a coronavirologist. So while I am going to be using SARS-CoV-2 as the case study for much of my talk today, depending on the questions that you have, I may point you to other experts. Okay, so where are we headed? The bulk of my talk is going to be this sort of boot camp session of molecular virology using SARS-CoV-2 as the case study. We'll talk about some general background. We'll talk about the infectious cycle, as well as how antiviral drugs work and some specific cases for SARS-CoV-2. Then at the end, I will talk a little bit about my own research, and I will leave some time for questions as well. So first, let's start with general background. Okay, so we're going to start at the very beginning. I put up this meme actually for my mom. My mom is a librarian, and she is, I believe, tuning in today. So hi, Mom. But in any case, at the very beginning, what are viruses? Viruses are obligate intracellular parasites. So let me kind of break down what that means. It basically means that for viruses to replicate, they need to get inside a host cell in order to replicate themselves. So that's what it means to be intracellular. And they need to use the resources and energy of the cell to replicate themselves, of the host. So they are parasites. One useful way to think about viruses is that they are really just molecular machines that exist to replicate their genetic information inside a host cell. So they get inside one host cell, they redirect the cell to make more copies of the virus, they leave that cell, they go on to infect more new cells. On the right over here is an electron microscopy image of a cell that's being infected by SARS-CoV-2, which are all of these little yellow dots, kind of a grotesque image. And one of the points I want to make from the top is that viruses are not sentient. Now, that seems kind of silly to say, but you'll often hear or read, or even I will say, that the virus wants to do something, or it confuses the host cell, or it recognizes something. But viruses don't want to do anything. They don't want to get inside this red cell here in this image. They're just these molecular machines. They're just a sort of a collection of dumb old molecules that are bouncing around until they find the right conditions that trigger the machine and initiate replication. That's essentially, at a very high level, what a virus is. Next big point, viruses are everywhere, and by which I mean literally almost everywhere. This is a slide that I've taken from online lectures of virology from Vincent Racaniello at Columbia University, and they are an excellent resource that I'll point you to. As he would say, we as humans live and prosper in a sea of viruses. So in fact, viruses infect pretty much every living thing that we know about: bugs, birds, fish, bacteria. If something is alive, there are viruses that infect it. Every day, we are eating and breathing and drinking billions of virus particles. And you can find viruses or evidence of viruses on essentially every surface on Earth. We even carry viral genomes as part of our own genetic material. So they're like integral to even who we are. Amazingly, though, we are not sick most of the time. And so the question is why not? Well, there's a number of reasons for this, some of which include we, for one, are not, quote unquote, good hosts for most viruses, so they don't actually infect us. The second is that some viruses have evolved not to create disease in us, so they might infect us, but they don't create any disease. And then the third is that we actually have remarkably good immune systems at many different levels. This is not how the immune system works, but I thought it was kind of a cool graphic to put up here. In any case, I would say that in many ways, viruses that cause disease, like SARS-CoV-2, are kind of the rare exception. Most viruses do not cause disease in us. Next big question, how do viruses like SARS-CoV-2 get into humans? Well, this typically occurs by what's called spillover from another species. So this graphic shows the likely origin into humans of various coronaviruses. So all of these little COVs are different coronaviruses. And as you can see, SARS-CoV-2 originally comes, the evidence very strongly points to the fact that it comes from bats, with maybe an intermediate host along the way, like a pangolin. And so what this means is basically somebody at some point had contact with a bat or a pangolin that had SARS-CoV-2 or something very similar to it. That the human then contracted the virus from the bat or the pangolin or whatever, and then went on to spread that virus to other people. So as kind of an editorial comment, what I would say is that at some level, the sort of pandemic that we find ourselves in currently, sort of spillover from an animal host, is really something that's inevitable and will probably occur again. So humans are spread across the planet. We're coming into contact with all sorts of wild animals, which means that we're exposed to their viruses. And it really is just a matter of time until one of those viruses meets all the criteria that to both cause disease and also spread well amongst humans. And then it's very easy to get a global pandemic. So that's kind of a depressing thought. But I mention it to really underscore the idea that we really need continued and more funding for basic and applied scientific research so that we can be better prepared for the next pandemic, which will come at some point in the future. Moving on, what do viruses look like? Well, they come in all sorts of shapes and sizes. Here are a few schematics of some different viruses, so influenza virus, adenovirus, bacteriophages, which infect bacteria, VSV, Sendai virus is one that we work on in my lab. At the center of each viral particle, I'm going to kind of use Sendai virus here. At the center of each viral particle is its genetic material, and you can see that kind of drawn as these little squigglies here. You can see it also in the influenza virus particle. You can't see it drawn for adenovirus. It would be inside this capsule. But this genetic material, it could be RNA, it could be DNA in a variety of formats, but however it exists, it contains all of the information about how to make all of the pieces that make up the virus. Surrounding the genetic material is what I will call kind of a structural housing. And this protects the genetic information of the virus as it's transferred between hosts. This housing will also help the virus get into the next host cell, as we're going to see here soon when we talk about the infectious cycle. As you can see, the structural housing varies from virus to virus. Some viruses, like Sendai and influenza virus, have a lipid membrane as part of their structural housing, and they have these proteins that sort of poke out of the lipid membrane. Some viruses, like adenovirus, do not and are just made up entirely of a protein capsule. Next question, how big are viruses? Well, we traditionally think of viruses as being very small, on the order of around 100 nanometers, which people always do this. I don't know that it helps, but it's on the order of 1/1000th of the width of a human hair. This small size is actually small enough that I put the word look in quotes. So the reason I put look in quotes is because 100 nanometers is actually smaller than the wavelength of visible light. So what something looks like at this size scale doesn't even really make sense as a question. We can quote unquote see viruses by using microscopes that use electrons rather than visible light. And then we get these grainy images such as this one of Sendai virus that was taken by one of my thesis students. So what about SARS-CoV-2? Well, this is a structural model of a coronavirus. And the key features that I'm going to point out is that the genetic material of coronaviruses are RNA. It has a lipid bilayer around it. So that's what this kind of pink thing is in the middle here. And at the top, poking out of the viral membrane of various proteins, including the spike protein, which is going to be very important for us going forward. On the left over here, you can see some early electron microscopy images of SARS-CoV-2 that were taken from a patient. I believe this was in January that this was published. And you can see that these sort of spikes, if you squint a little bit, that are poking off of the virus. In some images, they sort of create this crown around the virus. It's kind of what it looks like, which is hence the name coronavirus. Now, this sort of viral structural information is super important because it gives us insight into how the virus can operate, as well as ideas about how to treat infection or to destroy the virus. So just kind of as a basic example, understanding viral structure helps us understand how disinfectants work. So when you wash your hands with soap and water, or if you use a hand sanitizer, the chemicals are disrupting the viral structure. So the soap or the chemicals in the disinfectant. And what they're doing is they're dissolving the lipid membrane and causing the viral proteins to denature. This doesn't necessarily destroy the genetic material, which is kind of the heart of the virus, but it renders the virus non-infectious because without this structural housing around it, the virus can't get inside a new host cell. So viral structure is very important. All right, let's move on to next talk about the infectious cycle. But before we do, I'm actually going to do a quick sidestep and give a refresher about the so-called central dogma of molecular biology. I'm assuming that at least some fraction of this is probably familiar to most of you, but let's maybe dust off some cobwebs in case it's been a while. The central dogma says that DNA, among other things, contains information about how to make proteins. And proteins are the workhorses of the cell. They carry out most of the chemical reactions and do much of the structure of the cell as well. To get protein from DNA, the DNA is first transcribed into messenger RNA, and the central player in that process is an enzyme called RNA polymerase. It will read one of the strands of the DNA hybrid, and it will produce a complementary new strand of RNA. And this occurs inside the nucleus. This messenger RNA is then taken outside the nucleus into the cytoplasm, where it is translated into proteins by the ribosomes. And the ribosome basically just links up amino acids that correspond to each triplet set of nucleotides in the messenger RNA. So that's pretty much all I'm going to say on that. Hopefully that kind of dusted off some cobwebs. The big point in how this relates to viruses is that in many ways translation is the central process that the viruses are quote unquote stealing from the cell. They're going to make the cell ribosomes translate viral proteins instead of making cell proteins. Okay, so with that background, let's move on to discussing the infectious cycle. The infectious cycle is exactly what it sounds like. It's the process of a virus getting inside a host cell, taking it over, making copies of itself, and then those copies going on to infecting new host cells. Each virus has its own particular infectious cycle. Here I'm showing a schematic for SARS-CoV-2. So the red structure up here is the virus. The green sort of oval down here is meant to represent the cell that it's infecting. And all of these different numbers are steps along the way that we're going to discuss here in a second. Before we dive into them, however, I want to sort of point out two things from the top. The first is that only cells that permit all of these steps are suitable to serve as hosts for the virus. So if any one of these steps that I'm going to talk about is blocked or not allowed by the host or by the cell, then it cannot sustain infection by the virus. The second point is sort of a very general one, which is to not confuse the model for reality. This is something that I sort of try to hammer into my students over and over again. So the graphic that's shown here is the infectious cycle of SARS-CoV-2, and it seems very simplistic. There's sort of these 6 general steps, kind of migrates through the cell and generates new viruses on the other side. But reality, of course, is always more complicated and messy. To emphasize that, I've shown this really cool graphic by David Goodsell, who has tried to depict the actual sizes and concentrations of molecules inside a cell, inside a eukaryotic cell. So this yellow thing at the top would be the cell membrane that's shown out here. And whereas in our simplistic model, the cell is shown as mostly empty space, here we see that it's just jam-packed with proteins and other molecules. It's very chaotic. So I kind of want you to keep this in the back of your mind with pretty much all of the pictures that I'm showing you. Don't confuse the model for reality. Okay, so now let's talk about step 1. The first step is viral binding. And so remember from the top that viruses are just sort of these molecular machines. They're bouncing around until they encounter the right conditions that will trigger the virus to infect. So the first thing that needs to happen is it needs to bind to a cell that could sustain infection. binding typically occurs through a viral protein binding to a molecule that it recognizes on the host cell membrane surface. And this molecule is called the viral receptor. In the case of SARS-CoV-2, the viral receptor is a protein that's called ACE2 with another protein that's called TMPRSS or TMPRSS that is also involved. So you remember the spike protein that we were talking about before. The spike protein is the viral protein that binds to ACE2 and interacts with TMPRSS. And we can see a structural model of how those two proteins might interface if you're familiar with looking at ribbon diagrams of proteins like this. So what is this ACE2 protein? Well, it stands for angiotensin converting enzyme number 2. And what it does is it helps modulate blood pressure inside the body. So that's actually kind of important to emphasize. ACE2 does not exist to serve as a receptor for SARS-CoV-2. It has its own job to do inside the body, and the virus has evolved the ability to bind to ACE2 because cells that present ACE2 are good candidates for viral replication. Now, knowing what the viral receptor is super important because it, among other reasons, it lets us know what types of cells might be susceptible to infection inside the body. So ACE2 is commonly on cell membranes of cells inside the lungs, which is in part, not the whole story, but in part how SARS-CoV-2 is a respiratory virus. ACE2 is also on cells in the arteries and in the heart and in the kidneys and in large intestine or in the intestines, which means those could all be potential sites of infection for the virus. Receptor binding also helps determine or can help determine which organisms can be infected. So bats, for instance, have their own version of this ACE2 enzyme, which makes sense since the virus appears to have originated in bats. It needs to have a similar receptor that it could bind to. I'll also mention ACE2 just briefly in the context of news that you may have heard about certain blood pressure medications. So some blood pressure medications may actually result in more ACE2 being present on the surface of some cells. And there's been some worry that that might lead to increased susceptibility to infection by SARS-CoV-2 because if you have more receptors, then there's a higher chance that the virus could bind to those cells and infect them. In theory, that makes sense, but remember that reality is always more complex. So far, at least, there really hasn't been good data showing that, at least to my knowledge, that there's any increased risk of infection if you're using these blood pressure medications. Certainly not that way the benefit of taking those drugs. So pretty much every major medical organization recommends that you continue to take these medications if they've been prescribed to you. Now, because I'm a chemist, I also need to throw this in. What causes receptor binding? So in other words, kind of how does the spike protein recognize this ACE2? Well, I think, recognize, but of course, we remember that viruses are not sentient. They're just these dumb old machines bouncing around waiting to bind to their receptor. So what I kind of mean by recognition is that the viral spike protein has evolved a surface that matches the shape and chemistry of the receptor molecule. So this is kind of showing this generically for two molecules binding together. So the surface of the spike protein might have a positive charge where the receptor has a negative charge, or you might have these greasy nonpolar groups that are next to each other. But basically, the idea is that they more or less kind of fit together like a lock and key with a whole bunch of different magnets. And so when they get in the right orientation, they bind. And at a very high level, that really is all there is to binding, both in the case of receptor binding and pretty much any other binding interaction that's occurring inside the cell. Okay, so enough about receptors. Let's move on to step 2, which is viral entry. The basic idea is that after binding, the virus needs to get its genetic material inside the cytoplasm, which is where the ribosomes are. If it's just stuck to the outside, then it can't replicate. Now, there are many mechanisms of how viruses get their genetic material inside, but for SARS-CoV-2, the predominant mechanism appears to be through the process of endocytosis followed by membrane fusion. So let me talk first about what endocytosis is. Endocytosis is a natural process that happens in cells all the time, and it's basically a way for the cell to sample chemicals in its environment and maybe reuse them inside the cell. And the virus basically co-opts that pathway to get inside. Now, under normal circumstances, the way that receptor-mediated endocytosis works is that something, let's say this little blue guy out here outside the cell, will bind to a particular receptor, let's say these little red guys, and that will trigger the cell membrane to kind of bud inward, and eventually it will pinch off to form an isolated vesicle that's called the endosome. Inside the endosome, there are these little pumps that basically make it more acidic, so they lower the pH. And at the same time, there are other proteins called proteases that are delivered to the endosome, and they kind of chop up everything that's on the inside. So in many ways, you can think of the endosome as kind of a tiny stomach inside the cell. Now, normally, everything would get processed and chopped up into little bits, and then the bits would get recycled for use inside the cell. SARS-CoV-2, however, co-opts this pathway to get its genetic material inside the cell, or inside the cytoplasm. So the way this works is SARS-CoV-2, when it binds to ACE2, that can subsequently trigger endocytosis, so it gets brought into the cell and encapsulated by an endosome. And inside the endosome, when the pH is lowered and those proteases begin chopping up the proteins, as that process starts, instead of destroying the entire virus, that actually triggers the virus to inject its genetic material inside the cytoplasm. And it does that by the spike proteins actually mediate the merger of the viral membrane and the endosomal membrane. So the membranes fuse together, so it's called membrane fusion. And that membrane fusion is then what allows the RNA to get inside the cytoplasm. So that was kind of a lot, but at a high level, it's this process of endocytosis followed by membrane fusion, which is how SARS-CoV-2 gets its RNA inside the cell. One important thing just to remember for later on is that the lowering of the pH inside the endosome is an important trigger for membrane fusion, and this is going to be important when we talk about hydroxychloroquine. Okay, so now that the genetic material is inside the cytoplasm, replication can begin. And this has sort of two branches that are shown here, numbers 3 and 4. The first is that the host cell ribosomes need to make viral proteins. And these viral proteins could be part of the structural housing to make new viral particles, or the proteins can do what I will say is other stuff, which could mean that they could help replicate the viral RNA, so they could be polymerases, or help with that, or they could help the virus evade the immune system, which I'm basically not going to talk about at all. The second major branch is that the viral genome itself needs to be replicated, so you make many copies of the viral genome. In the case of SARS-CoV-2, there are a few sort of additional details that we know so far. The viral genome is RNA that can actually be directly translated by the host ribosomes to make proteins, so it can directly be translated. But those proteins are actually large proteins that need to be chopped into smaller proteins in order to be functional, and they're chopped up by these proteases. So that's not really shown here. But those smaller proteins then actually help replicate the viral genome, which is why there's sort of these arrows going up here. And they also then help generate other structural components, such as the spike proteins and so on, that will form the capsule around the virus. So at the end of the day, it's sort of this complex feedback loop that has redirected the cell to make all of the viral components that the virus is going to need. Okay, so now let's talk about these final steps of the infectious cycle, which is kind of self-assembly and then budding out of the virus. So after replication has gone on for a little while, the cell has made many copies of all the viral components, so the spike proteins and the viral genome. And after a while, you get a buildup of all of the viral components. And eventually, there's a high enough density of all the components that they begin to bump into each other, and they bind by that same chemical and shape complementarity. They bind and they self-assemble to form new viruses. In SARS-CoV-2, the self-assembly process occurs inside these vesicle membranes that are on the inside of the cell, and it will generate a vesicle that encapsulates a virus. It'll actually look very similar to the endosome. It's actually called an exosome. So this is exocytosis is what's going on here. But basically what happens is the virus is encapsulated inside this type of vesicle, and then the vesicle gets transported to the surface of the cell, fuses with the cell membrane surface, and delivers a new virus particle to the outside. Now, one big idea is that for every one virus that gets in, if replication is successful, many, many, many viruses will get out on the other side. So you get this amplification of the viruses. And of course, as the viruses leave, then they can go on to infect new host cells. Now, one last thing I'll mention here is that in many instances, once there's been enough production of the viral components, things sort of get so out of whack that the cell itself actually gets destroyed. And that could be one component of how viruses can cause disease, although not the only component. Lastly, let's talk about something that was not shown in that diagram, which is how the virus could then infect new host organisms. So we said that with each cell that is infected, you get many progeny viruses that are produced. And in the case of respiratory viruses like SARS-CoV-2, eventually enough virus is produced in the lungs or the airway that when a person coughs or sneezes or talks, there's a decent amount of virus that gets shot out of the person's mouth as these in droplets or aerosols. And that's actually very important. The virus, Sendai virus anyway, not Sendai virus, SARS-CoV-2, cannot survive on its own in air. It needs to have liquid around it. Now, if those droplets, so there's going to be a bunch of ifs here, if those droplets or aerosols make their way into another person's airway, and if there's enough virus that sort of survives the journey, and if the person has all the right cells in the right place, the virus binds to them that has the receptor, and if the virus gets past all of the immune defenses that I haven't really talked about, then finally, there's a chance that the virus could infect some other person. So there are a lot of ifs in that statement. But with viruses, it's really all about the numbers. So they produce so many progeny that if the vast majority of them never infect a new host, it really doesn't matter, as long as at least some of them do. So it's really all about the numbers. Okay, that's end of infectious cycle. Let me talk briefly about antiviral drugs and how they work. The high-level idea here is actually pretty simple. Basically, antiviral drugs are molecules that block one or more steps of the infectious cycle. And this is one of the reasons why understanding the infectious cycle is extremely useful, because it gives us possible targets or drugs for therapy. So these are some of the steps that people have been working on to develop drugs or test drugs that might block SARS-CoV-2 replication. There are drugs that interfere with binding, so these could be antibodies. There's just recently announced a clinical trial for promising antibodies that might work on this. There are drugs that interfere with the fusion step of the infectious cycle. There are drugs that interfere with the proteases that chop up those big proteins into smaller proteins. There are also drugs that interfere with the replication of the viral genome, so they interfere with the viral polymerase. So far, at a high level, I would say in the case of SARS-CoV-2, none of the drugs work very well, if at all. At least that's to the best knowledge of the data that we have. That's not to say that we won't have good drugs in the future. but it takes a very long time to develop good specific drugs that are also safe to use. So it may be a little while. Now, because they have been in the news so much, I wanted to briefly discuss two drugs for SARS-CoV-2. One is hydroxychloroquine, which is a fusion inhibitor, and the other is remdesivir, which is which is a polymerase inhibitor. So first, hydroxychloroquine. You've probably all heard about hydroxychloroquine in the news. What I want to do is talk a little bit about its mechanism. So hydroxychloroquine is a drug that's been used to treat malaria for quite a while, as well as a number of other diseases. It has a number of different effects in the body, not all of which are well understood, but one of them is that it increases the pH in the lysosome or the endosomal pathway. So you may remember from before that low pH, kind of an acidic environment, is one of the triggers for the virus to fuse and deliver its genetic material inside the cell. So the thinking has been that if hydroxychloroquine can increase the pH inside the endosome, that maybe that will reduce the likelihood that SARS-CoV-2 will be triggered to deliver its genetic material. So fusion would be stopped, and then replication would be stopped. So this is not a bad strategy, and definitely worth investigating. But remember that we don't confuse the model for reality. We need data, of course, to demonstrate efficacy, especially in the complex environment of a body, not just a simple schematic shown here. The data on this drug so far has been controversial with respect to SARS-CoV-2 treatment. There was some early data with a small number of patients that suggested that maybe the drug might give some benefit. Then there was a much larger observational study that claimed that it didn't make any benefit and it might actually do more harm than good. However, that study was then retracted just recently, like within the last week, which kind of clouds the whole picture around hydroxychloroquine. Separately, there has been a good randomized study demonstrating that people who were exposed to SARS-CoV-2 and who took hydroxychloroquine as a preventative measure, those people didn't show any reduced rates of infection relative to people who took the placebo. But we definitely need more randomized, rigorous studies to be performed to determine whether it might be useful. It doesn't seem to be useful in terms of prevention, but it might be useful in terms of treatment. The other antiviral drug that I will briefly mention is remdesivir, which maybe you've also heard about. This drug was designed to inhibit viral replication in general by blocking viral polymerases, so blocking the replication of the viral genome. And the idea was that many viruses have very similar polymerases, and so you could use it as a drug against many different types of viruses. How it works is it basically confuses, in quotes, the viral RNA polymerase. So this molecule looks very similar to one of the nucleosides that are added into the new strand of RNA. It looks very similar to adenine. And the viral polymerase incorporates this into the new RNA instead of an adenine. But the extra chemistry, so here's my chemistry, the extra chemistry of this carbon-nitrogen triple bond actually prevents the addition of subsequent nucleotides, which basically means that the RNA polymerase stops early. It doesn't replicate the full genome. And because you only have partial genomes, not all of the information for the virus particle is there, and replication is inhibited. The evidence so far is that remdesivir does have some benefit for SARS-CoV-2 infection, but it's marginal. So time to recovery in patients that use this went from 15 days to 11 days. It's an IV drug, so you get inside the hospital, so it kind of has some limited usage on that front, but it does show some effect. Okay, so this is where I'm going to end kind of the general virology section of the talk. If you find yourself interested in this topic, I would recommend these two resources. One is the online virology course that I mentioned from Columbia. This will give you 30 lectures about virology instead of one, if you're interested in that. There's also a podcast, This Week in Virology, which really is an excellent resource diving into the weeds in viruses. Currently, it's pretty much all about SARS-CoV-2, but if you're interested in viruses, these are awesome resources to check out. Finally, in the last few minutes here, I'm going to talk a little bit about one small project in my own research. So we're going to be shifting away from SARS-CoV-2, thinking about flaviviruses and Zika virus in particular. So this is work that I did mainly as a postdoc at Stanford and the University of Virginia. But then I'm going to briefly talk about work that we've built upon this here at Williams College. So the story begins with Zika virus that you maybe remember from the news about 5 years ago. Zika was identified in this forest in Uganda in 1947, and it was ignored for a long time because it didn't seem to cause severe disease. It's mainly transmitted by mosquitoes, so person to mosquito to person to mosquito. Like I said, it was mainly ignored until in 2015, there was a large outbreak that was reported in Brazil. And in that large outbreak that spread to other countries, there was an observed correlation and eventually a causation that infection of pregnant women could lead to neurological problems in the resulting child, especially microcephaly, which is this really just sad condition that you can see in a severe case that's shown here. Because of this, in 2016, the WHO declared Zika virus an international emergency, and of course it was all over in the news. That's kind of where I got involved. Zika virus, for kind of some additional background, is a member of the flavivirus family, which includes dengue virus and West Nile virus, which are both important human pathogens. Remember that I said that viral structure is important to know. So these are viral or structural models of flaviviruses that are based on electron microscopy data. They're about 15 nanometers in diameter, they're spherical, and they have this coat of proteins that is kind of arranged geometrically on the surface. This is a cross-section of the schematic of a typical flavivirus. So these are the proteins that are arranged on the outside. Underneath that is a lipid membrane, and at the very center is RNA, similar to coronavirus or to the coronaviruses. So that's kind of a quick primer on Zika virus structure. We were interested in studying questions relating to the early mechanisms in the infectious cycle, specifically the membrane fusion stage. So this is kind of a model that sets the stage for our questions and probably looks a little familiar at this point. So this is Zika virus. This would be the cell membrane, and then the green part here would represent the cytoplasm inside the cell. So the virus binds to a receptor, which has been sort of a controversial topic. There are some different candidates that are proposed, but it's unknown. It then gets taken up by endocytosis, similar to SARS-CoV-2, and then something is going to trigger it for fusion. So for other flaviviruses, the trigger could include low pH, just like for SARS-CoV-2. It could include these proteases that chop it up a little bit, also like SARS-CoV-2. And then there's also been evidence that it could include cofactors such as negatively charged lipids in the endosome membrane. So our question is, which of any of these factors might be true for Zika virus? And the first thing that we examined was low pH. So how are we going to do that? Well, we used a biophysical approach in which we could watch individual viruses fusing with artificial model membranes by fluorescence microscopy. So let me kind of walk you through this experimental design. The target membrane in this case is actually not a cell, but it is a membrane that we make ourselves, so we control basically everything about its composition. It's a vesicle that we tether chemically to a glass cover slip so that we can observe it on a microscope. This is all done inside a microfluidic device. Here's a real-life example of one. Here's kind of a schematic. But this allows us to very precisely exchange liquid in and out of the environment that the virus might experience. The virus itself, we fluorescently label with a dye that goes into the viral membrane. And then we basically just need to get the virus to bind to this target membrane, flow in low pH, and see if we can observe fusion, which could occur between the virus and the target. Now, of course, the problem is that we don't know what the receptor is of the virus. So how are we going to get it to bind? Well, we had previously developed a strategy for just this sort of situation, only we had developed it for influenza virus. And it basically uses these surrogate receptors that are called DNA lipids that we insert into both of the membranes. In any case, that allows us to bind them together, and then we flow in low pH solution with our syringe pump and see whether or not fusion can occur. So this is what the data looks like. Each of these little dots is a fluorescently labeled virus. Now, this is going to be a movie that I'm going to try and show. We'll see if it works. But at the beginning of the movie, the pH is dropped, and then we'll watch as these viruses fuse. You'll be able to see fusion because the little dots are going to light up. They're going to get brighter. So let's see if that will work. So we can see that as we go along, some of them, they start to get brighter, turning on like little light bulbs. Using this type of data, we can do some image processing and calculate the time between when the pH drop occurs and when the lipid mixing occurs, or when fusion happens. And from that, we then so we do that for one vesicle, or sorry, one virus, and then we then do it for many, many, many viruses, and we can construct something that's called a cumulative distribution function that basically shows us the fraction of fusion events that have occurred as a function of the waiting time. Now, the shape and time scale of this curve actually gives us insight into steps that might happen, intermediate steps that might happen in between pH activation and fusion. And we can determine that by mathematical modeling, which I'll discuss extremely briefly here in a second. But first, our question that we had talked about was, is pH sufficient to trigger fusion, right? What's the trigger for Zika virus fusion? And if so, across what pH range might it occur? This data set gets directly at that question. And there's really two things that I want to point out here. One is that on the left here, we see curves that were collected at different pH values. So up at the top here would be very low pH, so very acidic. Down at the bottom would be much less acidic. And the main thing to observe here is that as we decrease the pH, as it gets more acidic, we get an increased final extent of fusion. So more viruses fuse as we lower the pH. And this occurs in the absence of proteases or any other cofactors. And so it really directly answers our first question, right? pH is sufficient to trigger the virus for fusion, and it occurs across this pH range. So that was super exciting. The second thing that we observed was that if we normalized these curves, so they're all going to their maximum value here, we saw only a very moderate change in the normalized rate, or the curve shape, even across a really wide range of pHs. And this was initially very puzzling to us. You know, how can we get such a dramatic change in the final extent, but only a very moderate change in the curve shape? So to investigate that, we did some mathematical modeling. This shows the results of some very simple models, which are basically modeling the steps that occur in between, like I said, when the virus is bound and when it fuses. And in this case, the dashed lines are the fits of the model, and everything else is the data. Now, in the interest of time, I'm not going to dive into this too much, but suffice it to say that if we model the pH-activated fusion process as a linear process, so if there's many steps that can kind of occur sequentially, this does a very bad job of fitting the data. The only way to get closer to fitting the data, these aren't perfect, but better, is if somewhere in that pathway we include an off-pathway state. So something that doesn't let you move forward to fusion, but makes you go off pathway. So this was super exciting to us. You may be wondering why an off-pathway state is exciting. So there's two reasons. One is just sort of, I'm a basic science researcher. I'm trying to figure out how things work. So anything that tells me about how the virus works is inherently interesting. But second, and more broadly interesting, is that an off-pathway state could be a clear target for a drug therapy, because if we can stabilize that off-pathway state, then the virus doesn't proceed to fusion and infection is stopped. So we don't know yet what this off-pathway state is. I don't want to oversell where we are at, but this was very exciting to see this because it gives us an avenue to explore in the future. The last thing I'll mention is briefly this project, related project here at Williams with Abe Park, who's a student in my lab. And we really wondered whether this off-pathway state would hold true for other flaviviruses or whether it would just be true for Zika virus. So we got a data set thanks to a very generous researcher at Harvard University, Luke Chow, who was working in Steve Harrison's lab when he collected the data, and now has his own independent lab. And so he gave us this data. So all these open circles are the data. Again, not diving into it too much, Abe basically showed that sure enough, the off-pathway model, so all of these are the off-pathway model, fit much better to the experimental data than any of the linear models. So this seems to hold true for not just Zika virus, but maybe other viruses in the family as well. So there's a ton more I could say about this story. I'm super excited about it. We just actually submitted it for peer review, but in the interest of time, I'm just going to leave it at that. Okay, so what have we learned in these studies? Well, based on the data that I've shown you, I think we can make the claim that pH is sufficient to trigger Zika virus. Maybe other cofactors are needed, but pH is sufficient. And the pH range that it occurs suggests that it could occur early on during endocytosis. Modeling of the data showed that we seem to see this off-pathway state that's necessary both for Zika and West Nile virus. Going forward, we're really hoping to expand those modeling efforts and really develop even better models that might help give us insight into what that off-pathway state might be. Okay, so with that, I'm going to wrap up with acknowledgements and then take questions. Peter Casson was my postdoctoral advisor and really got me interested in studying viruses. On the Zika project that I talked about, I collaborated with Steve Boxer, who was my PhD advisor at Stanford, as well as with Beth Webster, who was just is an amazing graduate student and will be hopefully getting her PhD soon. The NIH supported the Zika virus work as well as internal funding at Stanford. Williams College supported financially the work on West Nile virus. And then lastly, these are the current members of my research lab here at Williams. Williams is a liberal arts college, so everyone in my lab is an undergraduate. I talked about Abe's project. Olivia just joined onto that project. I wish I could tell you about Amy and Nandini and Papa and Eunice's projects and other people that have been in my lab, but there really has not been time. But we have a lot of exciting and interesting work that we're doing. And also thank you, as I mentioned, to Luke Chow for the West Nile virus data. And with that, I will say thank you to everyone, and I would be happy to take any questions. And I don't know the process for this, so I assume that one of the panelists will hop in.
So this is Martha Meachem. We're having a little bit of technical difficulty in that we don't have a chat option currently. Although it may have just appeared. So if you see a little blue circle at the bottom of your screen that looks like a text bubble, now you can chat. Sorry about the fact that you couldn't do that during the talk. That was just a little error on our part, but we'd be happy to take questions in the chat now if you have any, although we only have about 5 minutes, so we obviously can't get to everything. So thank you very much.
Yes, thank you. That was incredibly entertaining. It was definitely a refresher for my biology skills.
Yeah.
We did prepare some questions while people chat them in as well. So I'll turn this Q&A part to Karen and Abigail, who will be our Q&A moderators.
Perfect.
Hi, this is Abigail. So just while we're waiting for more questions to come in from our attendees, I thought I'd start us off with a question generated by one of our panelists, actually. If viruses must disrupt cellular function to replicate, how could an organism be infected but not experience disease? Is it just that the viral load stays low enough?
Yeah, good. So there's, as with almost everything I mentioned, right, there's many, many, many more layers of detail and exceptions to various things. So there's many different mechanisms that viruses can infect and not necessarily create disease. One is that the virus could itself be latent, so it may not actively be replicating all of the time. One may be that it replicates at a very low level, so that it kind of goes on in the background and it's just kind of noise to the cell itself that's doing all of these other processes. Those would be sort of two mechanisms whereby it might not cause disease. Some diseases, I'll point out, are actually mediated, at least in part, or in some cases entirely, by our own immune response. And actually, in the case of SARS-CoV-2, one of the big hypotheses is that much of the severe disease is mediated by the immune response, not actually by the virus itself. So anyhow, hopefully that kind of gives some context for that.
Thank you so much. We have a question from a participant. Do you have recommendations to learn more about mathematical modeling?
Recommendations to learn more about mathematical modeling, yes. So there's many different ways of doing mathematical modeling, and I'll talk about the type that I know for my research. A good place to start is actually, we build a lot of our models based on chemical kinetics. So if you've taken a chemistry class, or you could look that up online, just search the term chemical kinetics. That will lead you to a lot of ways that type of modeling is done. This type of modeling, I'll point out, is very different than modeling that would, say, epidemiological modeling that would be done that generates, predictions of, infections across the nation or something like that. That would be a very separate type of modeling, which I am much less familiar with, so I won't talk about that. But I would, point you to any resource relating to chemical kinetics as a starting point to thinking about that.
Thank you so much again. We have another question from an attendee. For viruses that infect but do not create a disease state, might it be possible that there could be beneficial viruses similar to beneficial bacteria that live in symbiosis with us?
Yes. So this is sort of a more current field in virology of people looking at viruses that might be beneficial. There are some examples that I don't remember off the top of my head for organisms that are not humans that people have seen this. I don't know if there have been viruses that have been recognized, natural viruses that have been recognized as being beneficial to humans, but I'm sure that there are people that are looking for that, so it certainly is possible. I will say that certainly one of the tools scientists will use is they will actually engineer viruses to then deliver something that might be useful to the body or to themselves do something in the body. So whoever is talking about, there's a panelist talking about vaccines later on, and they may talk about this, but one of the vaccine approaches is actually to take a virus and engineer it so that it won't cause disease inside a person's body, but it will elicit an immune response, and that can serve as then a vaccine. So that's one way that it's not a natural virus, but we have repurposed a virus to be useful. Anyhow, hopefully that gives kind of some ideas there.
Thank you so much. I think we might have time for one more question. We have someone asking, can viruses be reactivated? For example, if a site in Europe is found where there are many buried people who died from the Black Plague, can that virus become active again?
Good. So could you get the resurgent of ancient viruses? Most viruses would not survive that long. So as I mentioned, for viruses like SARS-CoV-2, especially viruses that have lipid membranes, if they are exposed to air or harsh environmental conditions, their housing is typically destroyed, and eventually the genetic material inside is degraded. So anything that has existed for a very long time, the structural housing is almost certainly gone, and the genetic material is usually degraded to the point that it's difficult, that it would be very difficult for it to actually cause an infection. So usually, so the big answer would be no, you shouldn't be worried about that. In some instances, if there's well-preserved tissues, even still, the genetic information is usually destroyed beyond being useful, but there might be enough that scientists could take different sections of genetic material and reconstruct what the virus maybe looked like in the past. And this has been done to study viruses like the 1918 flu pandemic that existed long before we had the technology to look at them. So, so no, we don't need to be worried about getting infected from ancient viruses, but there can be interesting information that we can learn about how viruses maybe operated in the past in some instances.
Thank you so much, Dr. Rawle, for your time working on this presentation.
Yeah.
I want to be mindful of your time and everybody else's as well. And so this concludes the virology session. I do want to remind everyone that we are recording this session in case you'd like to take a look at it again. Apologies for the technical hiccups. So this is the end of the virology session, but we will now break for about an hour and return at 1 o'clock Eastern, 10 o'clock Pacific, for those of you over on the West Coast, for the next session on vaccines. Please feel free to stick around for this hour and watch the Tales from the Front Lines, which is the collection of recorded interviews from essential personnel working in public service positions during the lockdown. So thank you again, and see you all at 1 Eastern, 10 Pacific.